So I want to talk a bit about what we have been doing in the past about reconstructing branching and branching lineages from mostly single-cell RNA-seq data here. I will be given another talk on Friday in Aviv Reges seminar, or maybe something larger. I haven't quite understood the structure here, but that's going to be more biological. So here I try to be a bit more on sort of the algorithmic side, and I'd be totally happy if some of you would interrupt and ask questions in between. Particularly, I will be showing two new approaches we have been doing quite recently, and it'd be really great to get your feedback on that as well. So let me maybe start with a bit of an overview. So the big selling story of the Institute, I guess, is that, of course, we're interested in big data and life science across different types of topics, which kind of fits to, I think, what a lot of people, including here, of course, are doing. And I don't think I have to convince you that big, of course, in the life science case means it's not only the size that matters, but, of course, the complexity and all of this integration is, I think, maybe even more of a challenge than only the pure size. Although I think more recently, we being more on the sort of theoretical side also started to realize that it's important to actually be good also at implementing things, otherwise they don't scale, and it's going to become an issue in our field as well. And I think if you want to sort of give rough type of categorization of the type of data that you could start with, is of course a lot of sequencing and maybe multi-omics data that you want to integrate across multiple levels. My institute, we've been particularly working on combining genetics and metabolomics data in population cohorts, which is actually quite interesting because they sort of serve as an intermediate sort of phenotype, and you can actually learn a bit more about the disease processes. Then I think complex phenotyping is something that has been studied for a long time in epidemiology, but I think also in cellular assays it's becoming very interesting, and here I think particularly the Carpenter Lab has been working quite extensively on imaging and morphometry and so on. I think would be really interesting, and that might be one of the goals to discuss during my stay, will be to combine morphometry with actually single-cell RNA-seq readouts, and I think sort of copying ideas from epidemiology would be totally interesting to do so. Then I think bioimaging informatics in general is something that is sort of very well put towards the big data regime, because obviously you can do all the deep learning things very efficiently there because you have all these nice additional local structures, so you can make these convolutions and so on. And then this is something that's maybe not so common, but I want to put it out as also one of the increasingly big data sets. It's this it's single-cell profiling, either based on images or maybe also on RNA-seq, where you really start having data sets that are so large that you actually have to maybe do different things, and maybe you also can do different things. It used to be that, particularly in the omics case, we have been on this small n large p problem where we had so many high-dimensional parameters and so few samples. I remember still getting the first 3 microarrays or so 15 years ago and trying to do something on that. Obviously, you had to regularize super strongly, and now we're getting in a different regime where you can actually do more. And just as one example, what we have been doing in the past was, for example, we tried to build up a risk score for type 1 diabetes, a disease that's strongly known to be hereditary, so it has to have a great genetic impact. And in this case, what we just did, we had a test cohort, we validated on a smaller one, and we just combined essentially logistic regression models. We just basically combined a series of SNPs in a weighted fashion, learned the weights, and did some type of feature selection to be a bit fancy, did a Bayesian version of that, but essentially, I guess Lasso might have done the job as well, not so much worse. And we got quite good prediction accuracy, and I like to show this story not because it's a super complex model, but this is really something that has been actually taken up by the medical doctors, and we have a collaboration partner. She has started a cohort study, type 1 diabetes screen in Bavaria, where she's actually using this risk score as a secondary screen for picking up a risk of newborn babies for type 1 diabetes. And sort of from the health perspective, this is super useful, because if you know that you have to be careful with your baby, you start maybe treating it a bit more carefully. Yes?
So is this a polygenic risk score based on common SNPs or rare SNPs or both?
So it's a polygenic risk score. It's really based on common SNPs.
So it's the standard GWAS approach plus some Bayesian regularization?
So you get the risk SNPs from a GWAS, but now you combine them. And traditionally, particularly in the field, the polygenic risk scores have been always combined with fixed weights based on an frequency or something like that. Yeah, and we train
based on the effect sizes from the GWAS.
Oh, effect sizes, yeah. And in this case, you can show if you adjust the beta. So if you really train the beta, so you get actually much better prediction quality.
Oh,
which is to be expected, right? Because you don't have interaction terms before.
that's true.
So, and you can also test for interactions. You can see if something nonlinear works and so on.
Yeah.
Turned out not so much.
Yeah, that's always the story of humans so far.
But what was nice, I mean, in particular, there's one locus called HLA, which is dominant in this, and it was still only a minor improvement above HLA 0.05 or something like that, but still was. So it's actually that was nice. And if you want to do the same thing, our center is interested in diabetes. If you want to do the same thing for type 2, for example, it's actually not really worthwhile to look at genetics, because there, I mean, you just look at, I guess, age. I hate to break it, gender, of course, males get it more frequently, and then I think body mass index, and that's pretty much about it, right? So there, genetics doesn't help at least for the prediction. But this sort of this prediction is something that you can do with large data sets, and of course now here more complex phenotyping and so would be of interest. But what I want to do, and this is sort of the promise of data science, of course, you can replace pretty much straightforwardly sample person by sample cell and actually do similar methods on that field. And for us, it turned out to be really fruitful and productive to use a lot of tricks from epidemiology to actually analyze single-cell profiling data. I just want to give you a few examples in a minute. I want to work on single-cell RNA-seq data for now mostly, although we have been also working a lot on imaging data. And just to say that this actually goes now towards a regime where the size is actually really getting large, here's a sort of traditional development of single QRT PCR data sets that started becoming sort of, at least in the range of a few thousand of cells, but obviously the number of parameters was limited, similar to FACS assays. And when you, with CyTOF, actually you get maybe into the 50s or maybe even 100 or so of different parameters, but to be really genome-wide, there has been ideas such as Murphy's, very interesting also with respect to spatial localization. But with RNA sequencing, in particular now drop-seq or in-drop, you actually get to regimes where you have many cells as well as many transcripts observed. So you get to this many gene, many cell regime where you can actually do maybe novel things such as this lineage estimation. And just to point out that this is actually also a nice playing ground for people that maybe don't want to sort of collaborate deeply. There's actually a lot of data sets that are being published now over time in the field. In particular, the number of cells in each of these data sets is totally exploding. I think while one of the first papers has been published, very high impact, I think with 8 cells or so. It was very efficient sample use. I think nowadays there's data sets around. There's one from 10x that has more than 1 million cells. So 1 million of transcripts that you have, millions of transcriptomes that you have to analyze. Obviously, it's super shallow sequenced and super noisy and so on. So you have to, on the one hand, you have this nice power of high number of samples. On the other hand, of course, you have also the challenge of dealing with all of this noise and maybe going beyond sort of a very standard noise model. So it's a nice opportunity, but also gives maybe a reason for taking up again questions that should have been answered before. For example, at a recent single-cell conference in Kingston, I think half of the methodological talks were looking again at clustering, something that you think should have been solved 20 years or so ago, but it turns out, well, you have to maybe do something a bit more special there. So what's the general idea of such an analysis? And here I took the slides from a recent review from Rick's group, where the promise is I think quite obvious. If you have some tissue that is heterogeneous and you want to compare it to a pathological situation where maybe there's some subcell type that pops up potentially in a rare manner, and you want to pick up what's different there. If you were only to compare the averages, you might see slight shifts in differential expression of a few genes, but you might not actually learn much, particularly if the number of cells here is very low. So the idea would be, if you can really do this on a single cell level, you would actually pick this up more easily. So you do this single cell RNA sequencing. I don't say anything how this works. I mean, I think the idea is clear. You have to have a very good amplification protocol and a very good cell isolation protocol. And there's lots of protocols around, and they're being improved. But I want to focus on the analysis now, right? So the idea is that is so very nicely put into expression profile clustering, and then you end up with what we call cell type maps, which would be sort of a low-dimensional visualization of similarity of those cells. So basically, each of these different colors of cell types have different profiles, and you want to aggregate them now by similarity, you get these clusters, which would be cell types, and you maybe have a nonlinear dimension reduction to show how they align, and then this small cell type here pops up. So this sounds like a straightforward analysis pipeline, but of course in practice there's all kinds of problems, and maybe just to point out a little bit what the computational challenges would be is even just starting here with the pre-processing. There's a series of just basic QCs or so that are not as yet worked out as they are for bulk RNA-seq, and particularly with each of the new protocols you have to deal again with, I don't know, amplification bias, something with the bar coding. So there's a lot of new questions that you could ask there. Then this visualization is something we have been working on a bit. So this dimension reduction from 10,000 or so to 2, where PCA might not just cut it, you have maybe missing values. In addition, you have this a strong zero inflation because of dropout and also just SAMs not being present. So you actually might want to adopt your dimension reduction technique there. Then this clustering, as I said before, that's actually really something people still ask quite a lot. And then also just differential comparison is something that on the bulk level, of course, essentially boils down to a fancy version of a t-test, right? You just want to know, is this across your replica, is the distribution of one gene the same or not? In this case, actually, you might have a situation where you have the same mean, but the variance, for example, might go up, right? And this would actually tell you something about the heterogeneity being increasing in your population. So a standard t-test or whatever more fancy version you cook up in DSEQ2 or so, this doesn't cut it anymore. So you would actually want to test for differences maybe in the tails, or maybe also more complex, of course, you want to maybe just test for differences after the clustering. So again, people are also starting to ask how to do better differential comparison. And I think in the MIA, you recently had Peter Kachenko also give a presentation, right? And he was one of the first to actually ask this, I think, 3 years or so ago already with the SCDE, and he kept improving this. And I think he should have presented Pagoda, I guess, right? Yeah, he did. So you saw that there's sort of new noise and models coming up for that. And at the very end, I want to briefly tell you something that we have been starting to do, where we basically combine this one with clustering, trying to work out better differential expression. Be happy to get feedback there. And then, of course, all kinds of downstream analysis. So once you have processed it, you maybe know what's differential. One of the big promises, of course, sort of what I think has been driving the field for quite some time is, can we maybe not only find clusters of groups, which most of the papers, maybe up to maybe last year, have been mostly doing new tissue. Oh, I found heterogeneity, I find cell type ABC. But it would be actually interesting, what type of genes are doing something differential? What type of maybe even regulatory programs are going on? Can I learn something about mechanistic gene regulation? Could I, for example, take the heterogeneity that I see here, this natural fluctuation, to correlate genes across cells to really get regulatory networks out? Maybe not only by correlation, but really going towards full causal analysis and so on. And I think these data sets, let's say, if they have been properly cleaned, could really help us answer these questions. And before I tell you what we do on downstream, let me just show you two or three brief slides on pre-processing, also because I want to sort of get the bracket to the epidemiology. So my lab actually started working that with a talented postdoc in the lab, Florian, where we initially just worked on qPCR, single-cell qPCR data. In this case, for example, we looked at blastocyst development, where we had 2, 4, 8, 16-cell stage, and then saw sort of split off into different regions, and essentially came up with a Gaussian process latent variable model that was adapted to this particular noise situation to visualize this type of data sets. It's similar. We looked into qPCR data and saw that you don't have only these nice normal distributions. So here we just plot across all genes cycle time values. So it's essentially gene expression, and we saw then these strong peaks here in the data set. In the original paper where we got the data was from the Robson lab about this developmental data, they actually just did PCA on that, and it kind of worked. But of course, you know, right, in PCA, we essentially make the assumption that we just look at the normal distribution, and we want to sort of see where we see strongest variance and project along that, right? Turns out, if you have such big things here, you actually find then stripes in the data which come from this. I mean, this is what in RNAseq is called zero inflation. So this comes from the strong non-expression of genes, let it be dropout or just not being expressed here. So this bimodality actually brings some structure in your data that might not actually be present. What happened here was that simply in the qPCR protocol, they just stopped measuring after 25 cycle times. So they just set everything that was above 25, they set to 25. So in fact, in statistics, it's called censoring, right? And you should adapt your method to deal with censoring. So what we did in that paper, we essentially just had a PCA with a probabilistic PCA with a noise model that was taking into account this censoring effect, and then you could basically smooth out this thing, and then you didn't find this type of structure. And maybe the latest thing that we did is this was a collaboration with the Stiegler and Marioni lab that Florian visited at the time, was that we started realizing that when you look at bulk data and you typically have asynchronously differentiating cells, you sort of average out very nicely cell cycle effects, right? Whereas if you look now at a single cell and say it's early and the other one is maybe late during the cell cycle, the late one should have on average twice as many RNAs as the one in the beginning if it just wants to maintain its level, right? So by sort of not taking into account this growth, you would have a smearing out by a factor of 2 of all of the expression values. So what we did, we just looked at a GO category and maybe a bit more fancy version of cell cycle genes that we reduced to a one-dimensional factor that we then regressed out of the original gene expression. So similar, and this was motivated by epidemiology, where if you want to compare a patient versus control, we also often regress out, I don't know, gender, ages, or things like that. So here we just regressed it out in a random effects model, and then this essentially corrected for this effect, yes.
So do you provide cell cycle genes a priori, or you discover them as well?
A good question. So in this case, actually, in the first iteration, we really fixed the model to known set of genes. In this case, cell cycle genes. Also, we then had another GO category for differentiation. We wanted to see how strong that effect is. But in this case, you had to fix it. So Florian then moved on to do a postdoc at Ollie's lab, and I think I don't know if it's already out or it's in press. He called it the SCLVM or something like that, where you can actually also detect an unknown category that does this effect. And I think Pagoda has something similar also included, yeah.
So it was a linear mixed model?
Yeah, it was a linear mixed model.
Cool.
But with a random effect. So in the later one, actually, he starts doing a fixed effect because it turns out this is computationally a bit demanding. Yeah. So, I mean, Ollie has this Linux package that is rather efficient, but still it's... So in practice, for many situations, it's also okay to just do a PCA on that and regress it out, right?
That's fixed effects.
Yeah. Okay. So in practice, very often, this is just faster, so we often do it, yeah. But maybe just to point out, yes, oh yeah, sorry.
I just wondered about times when you have cell cycle effects that are really confounded with actual other effects that you're interested in, which is frequently the case.
Yeah.
So then it would be more like all of your men have the disease and all of your women don't. And if you were totally just regress out men versus women, you also
you don't see effect,
right? The other effect that he's doing. So, but you're not necessarily interested in discovering only the cell cycle genes, which are very obvious to see often. So, is there some kind of hierarchical thing you could do, or have you looked at all of these?
Yeah. That's a very important point, right? Very often, so particularly when you discuss this with biologists, say, yeah, but I'm interested in cell cycle because I know that it affects my differentiation or whatever, right? You know.
Or you're not interested, but you know it's really confounded with...
Oh, it's confounded at least, yeah. So what we did, in fact, we essentially extended the linear model by not only saying we have one covariate from cell cycle, but we have another covariate from differentiation, then we have a product that's a sort of an interaction term between differentiation and cell cycle, and then you could try to fit this more complex model. But of course, in particular with this random effect model, this fitting is not as robust, so it's a bit of a beast to deal with that. And I think in practice, what many workflows nowadays do is they try to quantify the effect of that, but then you might want to maybe work on this sort of on this subset separately. But you should always sort of whenever you do some type of reduction, then plot and then you see how homogeneous all your cell cycle contributions are with respect to your condition that you have, because you typically know about this condition, right? It's a very good point. But let me just say, you know, initially I kind of suggested that it has been, one of these big ideas has been to go towards correlation networks and learn something about gene regulation. And if we do this with the cell cycle effects, and in this case, this was supposed data from Sarah Teichmann's lab, Fluidime still, where we had naive T cells differentiated to I think Th2 cells that are sort of known to be strongly cycling, and we get this correlation matrix during this differentiation process, 200 cells or so. And then you know, if you see, so this is just genes by genes, right? So you see there's a strong set of genes that are upregulated. You would kind of interpret this as a regulatory network driving potentially that process. But turns out that most of this variation was actually used by cell cycle. And if we remove that effect, we actually just got this small set of correlated genes. And in fact, those really correctly enrich for GO factors that are related to T cell differentiation, whereas this one is really just dominated by cell cycle. So in very many situations, cell cycle is sort of masking maybe some more subtle effects. So, you know.
And masking it in a way that a naive fixed effects model would overcorrect for, maybe, right? Or do you think it's fine?
So I don't know if we tried it on this situation. In practice, very often this is also, I guess, okay. But I mean, in this case, we just didn't correct it all, right?
Sure. Yeah. It feels like there would be more risk of what Sam mentioned, which is that you would also wash out things you're interested in that correlate.
Yeah, true.
Or,
yeah, I agree. So and now let me show one more pre-processing thing that we're currently developing. So this is not yet done. This is a collaboration with the Teichmann lab, where we started realizing that because we want to upscale a lot of these analysis, we do more and more more batches at the same time, flow them together. Or let's say we have different patients and we want to integrate them and want to sort of analyze data across batches. So we started thinking we have to find a way of choosing one of an optimal batch correction methods. Even in bulk, I think as most of you know, there's many of those methods around, and it would be good to have some type of index to quantify that. So what we did is we tried to find some way of comparing well-mixed data with maybe a not so well-mixed situation, where now here the colors would indicate 3 different batches, and you see here there seems to be clearly some shift of the batches in this toy example to the right. And the simple idea we had to actually quantify this, so basically what people do at the moment, essentially they just do a PCA of the data set and see if the colors overlap or not. I mean, that's sort of, very simply put, the state of the art, I would say. I guess maybe they quantify also, maybe they quantify contribution of the batch effect to the PCs. But sometimes it doesn't matter all that much. If you do differential expression, very often you can actually account for batch effect in whatever downstream linear model you do, right? But for example, for some unsupervised things, very often this is not so easily the case, or it's not commonly done. So at least standard pipelines such as Surat and Skater, they very often do pre-processing to remove batch effects. But as I said, it's not clear what type of pre-processing to do. So our simple test now goes as follows. We just look in neighborhoods of each of the different cells, and we try to see if we find locally the same heterogeneity, so percentages of the different batches that we find globally, okay? So very, very straightforward homogeneity test. So in this case, right, they all look sort of homogeneous, whereas here this one clearly does not, so it seems to be enrichment for gray. This seems to be okay, but here seems to be enrichment for the blue, purplish type of cells, don't see colors. So that's essentially the test, right? So you compare the number of a label I in neighborhood K around a particular cell J with the frequencies that you know from the total data set. You normalize them, and then you know this is chi-squared distributed. And you do this now across different type of pre-processing. So we call this K-BET, which stands for K nearest neighbor batch estimation, I think. So we were discussing about the name, but Martin liked it. So I should say that's my PhD work, and this collaboration with Sarah and her postdoc Chichao. So essentially what we did, and this will sort of involve most of the work now, is that we actually now took raw data, look at different normalization strategies, different batch regression methods, and many out, I don't go into detail now, and then we evaluated them, as I said, with our index, and then we compared this to a few other indices, and we find very quickly many situations where visual comparison or just pure PCA-based comparison, where PCA sort of measures just dominance of the variance, just doesn't cut it anymore, right? And I just want to highlight this because I think a lot of you have heard that Aviv, together with Sarah, pushing this human cell atlas project, where we actually be starting to produce many of those data sets, and they're actually we're just planning a hackathon where we want to evaluate different of those batch regression methods. So I think it would be really good to have a robust measure. So if we do this measure, and now I show you just effect on one data set. This is from a recent cell stem cell paper from Sarah's lab, where we have multiple batches from mouse ESCs in different conditions. I just show you here, I think, for the 2i condition now, different batch correction methods versus different normalization methods with this particular batch measure, where now in this case red should be good. Understand this correct? No, I think so. So low k-bet is what did I say? I said it measures homogeneity. So low k-bet goes to 1 so high. So blue should be actually on the good side. Yeah, sorry for that. So now so we played around with it. We were quite happy with comparing. But of course, if you think about it, if you have a very strict batch correction method, let's say you have a batch correction method that maps everything to 0, then our homogeneity is very high. So of course, in order to compare correction of a method, you always also have to see how much of the original structure it pertains, right? So it's always a trade-off. And so just as well, we also now quantify in a second analysis how much we get overlap of original gene enrichment of highly variable genes in terms of gene ontology categories with this our particular measure. And as I said, I don't say now in detail what the different methods do, but now we can basically compare this overlap with the k-bet measure and be interested sort of in this high region where tools perform well. And in many situations for us, IUV, I think G correction seems to be doing a good job, but we can't give a general advice because that it does vary across data sets. So our current advice is basically use this to try, if you have time, try out a few and then do this analysis. Yes?
So this is cache, where you like do this.
Oh yeah, I didn't say that.
Is it designed for all the data set that you use, or did you verify this data set?
Yeah, that's kind of a problem that many, so I later show you methods that are based essentially on k-nearest neighbor graphs and to do all these graph type analysis, and sort of very often you don't really speak about it, right, like in PCA. In this case, actually, it's rather robust across K. So what we in fact do, we scan through all K and we typically get such a behavior. For very large one, it's degenerate, very low one, it's degenerate, but in the middle you get some plateau, and we have a divide and conquer strategy to sort of roughly get to the plateau. So in this case, it doesn't, because it's really, I mean, you average then over all of these local things, so it does not do so much. Yes?
So are any of your conclusions sensitive to whether the data set is from a protocol that has UMIs or it doesn't have UMIs?
Yeah, thank you for that question. This is actually, this is something we got a few times already. Can we make a recommendation for drop-seq versus, I don't know, fluid and so on? I think we looked at 5 or 6 data sets, and in most situations, IUVG, I think with a simple log count preprocessing was actually doing a pretty okay job, and other methods such as PAIR never did very well. Combat, I think, was also quite okay. So in these data sets, it turned out to be this case. I don't know if there's no way theoretically I can argue why this is so. Okay. So much for the pre-processing. So let me now review briefly what we have been doing on visualization. Yes, thanks.
If I could just ask, so that was kind of comparing similar samples, but where does one draw the line between batch and biology, right? Because obviously you can go back and say, okay, now I've regressed out my variance, cluster my things together, then you can go back to some hybrid state between your original and that to figure out where your biology lies.
It's a tough question. So basically, if I can reformulate just to see if I understood the correction, the question. What you're asking is, at what point do we correct too much? And this is sort of what we measure with this overlap, but also let's say you have two different conditions and there's only a subcell type responding, do you really then want to remove that effect? Obviously you don't, right? So you want to see this. So I think what we do for evaluation of these things, we actually try to go to as homogeneous as possible subpopulations to look for this effect. In practice, I think it's quite a tough situation. Very often nowadays, you actually really want to compare your heterogeneity across different, let's say, even patients, right? And we know that in patients, there's definitely going to be some type of effect. So, I mean, what we do, in the differential expression analysis, we always account for sort of the design, right? But usually just in a linear fashion. So, yeah, I'm not sure. So I think most of these batch correction methods actually have been proposed for the bulk case. So of course, a quite obvious question would be based now on a sort of local homogeneity measure. Could you actually maybe sort of keep pushing these batch correction methods as well? Ideally, you want to have some type of formal model of what actually generates the batch effect, but I don't think this is really realistic. That's why we are left down to playing. So yeah, I think it can't see more than.
Normalization is usually used also to account for how well the cell was sampled, right? Individual cell effects. And so are you saying that you don't do that normalization and that is better? Or, sorry, I just didn't understand. Because you're quantifying batch effects, but you're sort of mixing that with also normalizing for cell.
So we basically want to so what we want to compare is a batch correction method as well as normalization method. So we have sort of this matrix comparison of things. So we look at different types of normalization methods, and you're fully right. Very often just compare for, I don't know, this cell hasn't been sampled as deeply or something like that, right?
So but the methods that you ended up sort of preferring didn't seem to do any normalization. Is that what I understand?
No, I think the color indicates normalization. I just don't see color so well.
Oh, I see. Okay.
Sorry for that. So.
Okay.
So here we have basically comparison of each of those, and I just summarize this because this is so difficult to understand in this one, and I think this one should be log normalization. So yeah, so we need to do something, but I think scalar log account or something. So now I want to speak briefly about this visualization using random walks, and this is basically the idea that we use now to go to lineages. I just have to check the time that I don't go too much over it. Okay, so this is work by Nale, a PhD student in the lab who also recently moved to a postdoc in Cambridge, UK. And she's been interested in now comparing cellular profiles in situations where you don't actually have cells clustered up so well, where you actually have some type of developmental process, something like that going on. So we have this high-dimensional transcript space, and because we have now large data, right, we don't only have a few, but we have this high dimension, we have many of those cells. And ideally, in this 10,000 dimensional space, they maybe just don't form a big blob, but they might actually somehow reveal the structure of the underlying process. So they might look like, let's say here, this would be some type of progenitor cell, and then they go down, and they might be doing some type of splitting or something like that. So we somehow want to analyze now this type of transition process. And the idea that we do this is by looking at random walks transitioning through this high-dimensional transcript space. We basically just jump to nearby cells by putting some type of Gaussian kernel on these jumping probabilities, okay? And if you want to reformulate this, you could call this that you diffuse to close by cells with a high probability than farther away. That's why this technique is then later called diffusion maps. So the idea is you write down a cell times cell transition matrix where you have a probability of transitioning from one cell to another just proportional to a Gaussian kernel. So you get this Markov transition matrix. And then you superimpose all of these transitioning random walks. And this particular example that I showed you, then you see that you get a high density of walks going basically along these sort of lines that are connected by cells. So if you had now some means of visualizing that, you would actually see that there seems to be one dominant and then a second dominant axis. Yes?
So you calculate the distance by Euclidean distance?
Oh yeah. So in this case, initially we do Euclidean. But I didn't say something for single cell. For single cell, actually, we have to treat the zero inflation differently than the rest. Yeah.
And normalization obviously makes a huge difference.
Totally. Yeah. Very much.
And is sigma adaptive?
Yeah. So this is a very good point. Initially, of course, we didn't do it, so we just chose some rough sigma. But particularly in development, you end up having states where you have many cells clumped, and there's very few cells that are transitioned through. And if you don't do it adaptive, you basically just smear out these few ones. So we have a local density proportional inverse proportional sigma. So initially, just work on Euclidean distance. Actually, I will define a distance based on this diffusion map concept that I think is actually more adapted to this high dimension data, sort of more geodesic, I think, which we cannot show, but just experimentally show, but we're happy to discuss more about that. So, but what we do now is we take this Markov transition matrix. Yes?
Does this new distance that you mentioned take into account these dropouts?
Yes, so I have a slide on that. Just give me a second. They don't, so we say it does, but I think it could be improved. Okay, so it's, yeah, so we're getting there. So let me just, so far it's just really the standard diffusion map. So this is not our idea, it has been proposed by Kaufman already, I think 10 years or so ago. So what you do is you take this Markov transition matrix, and now you want to see what's dominant in this transition matrix. So what you do, well, you just take from your old linear algebra toolbox, you take what eigenvalue decomposition and see what's dominate, what's the first and second eigenvectors, and then you plot along these two, and you call those diffusion components. So just as where in PCA, you take the covariance matrix, here you take this transition matrix to nearby cells. And because you take this transition matrix, you have now a nonlinear version of that. So it's not a linear projection anymore. And in fact, for this thing, you see that the first DC is actually dominated by this process, the second one gives you that. So somehow gives you already something going along these lines. And what we did now in this particular paper, now we actually sort of looked at this old approach and did a few extensions. For example, we looked into more type of probabilistic interpretation of the method, so we actually had some type of account for dropout and uncertainty in this scaling parameter that we discussed. Initially, we did a global version of that, and then later we generalized this to a local version, where we essentially tried to quantify somehow this intrinsic average dimension that you want to take at a certain locus, and so you get a localized type of sigma. And as I said, yeah, this local sigma actually accounts for this sparsely sampled region. Still, it's a heuristic, right? So there's nothing. So we couldn't really show some type of asymptotics or something. So
sure. So locus. What does locus mean in this context? Use the word locus.
Local. Sorry.
Oh, okay.
Okay, so we just yeah. So okay. This means basically we look at some type of estimate of local density. Initially, we averaged to do some type of global estimate of sigma, but then later we did a localized version of that.
So I thought Quantifying's original work actually has exponent alpha, which, when treated, I think, as -1, already does this can be interpreted as adjusting for local density.
That would be interesting.
Okay, we can talk later.
Yeah,
There's different flavors of standard diffusion maps.
Right.
Yeah.
There's sort of one that's the Fokker-Planck version. There's one that's non-density adaptive, and one that is density adaptive, and they just differ in a certain exponent in the denominator of the.
You could sample non-uniformly, but if you sample nicely from like some submanifold and then noise out a little bit, then there's a theorem that you recover it even with totally non-uniform density.
Right. So you get a nice theorem in Riemannian geometry, actually, that you recover the measure as well. Anyway, we can talk.
Yeah, that'd be good to see. So I think originally when we looked at that, we went quite a bit through this. But
you might be doing a better job of correcting. With real data, you may have a method that is much better than what the theory says might work. I mean, that's very likely.
The theory also makes a lot of assumptions about the noise.
Yes. True. Yes. Yeah, the theory assumes all kinds of things about the noise that are probably not true.
I mean, in this case, because we have sort of this two-component noise, so we have this sort of nice behavior here, and then
there's not uniform on the manifold and off the manifold. And then you still have the zero inflation. Anyways.
The thing is, and this was what, because I think there was a question about the zero inflation, the dropout, how we keep it. So here we really just say that everything in the zero is dropout. So we treat this completely differently than from the normal distribution. What actually in practice you should be doing is that you should split up the zero component in one part that's just not expressed, another part that's actually coming from, I don't know, experimental average. So what people nowadays do is actually do some type of zero inflated negative binomial model. And we speak about that in a minute. So far we didn't do that for this approach. But I think it should be actually possible to extend. Yeah, and maybe the last thing I should actually stress, this took maybe most time, but turned out to be most useful, is actually to also have a large sample extension of that, because to do that, essentially you have to build this big transition matrix, and if you do this for 1 million or so cells, I said datasets become that size, actually it does not scale in its original implementation. So we actually came up with an implementation to have basically a nearest neighbor, a sparse version of that. Yeah. And you have to do some tricks to make it efficient.
Is that essentially thresholding?
Yeah, essentially thresholding.
It's a small 7 to 0. Okay.
So you're not.
But you have to have a good search to really determine which ones are close by.
Did you think of it? So you're not truncating the spectral decomposition of the IND?
This is something we do when it becomes very large to be approximate, but originally we don't yet. So in this case, we just really set things that are far away in this Markov transition matrix to 0. So that's the first. So that's 2 approximations. The second one is actually you do some truncation.
Low rank is different. Yeah.
It's true. So let me show you an example of what we did with that. Yes? Okay.
What was the question?
I think you asked if we do this on the full transcript space, or if we do dimension reduction, just look at highly variable genes or something like that, right? And this is, I think, for most of them. So, for example, for TSNE, very popular dimension reduction. Very often, a lot of those are very sensitive to what you actually throw in, right? And in practice, here we don't see so big of an effect for this transition processes, particularly if you want to reconstruct lineages. We do it for qPCR of the same system as well as for RNAseq, and if sort of qPCR selects sort of big dominant things, actually it turns out to be okay. But again, by theory, you would expect that input does matter. So I don't think I can give an advice. In practice, we usually just take a subset of variable ones. So let me show you an example that we did in a collaboration with the Göttgens lab from Cambridge, where in this case it's still qPCR data of 4,000 or so cells taken at various stages in embryo differentiation. The question was, in embryo development, the question was if we see at this head-forward stage a split into more blood-like cells and more endothelial-like cells. So they have been sorted at the time, and just by standard clustering approach, this is just with Euclidean distances, you see that you see one component here, so this putative blood component, they sort of look all the same. So here you have the different cells, here you have the different transcription factors that have been taken in that particular experiment, and you see some dominance from different factors. I think this one is HBBBH1, which is known to be up here. I think RUNX1 also pops up here. The rest, though, is a bit more salt and pepper, so you couldn't really see much structure. And of course, and I pitched the diffusion map idea, so we basically applied that to that, and suddenly it looks much nicer. So you actually really see this type of splitting process here in the data. So you see here early this primitive streak dominating, also already being mixed a bit with this neural plate. So from the particular experiment, we see that some cells seem to be already later and earlier in these stages, and you see some sort of split up into this more endothelial and this blood part. Now you can superimpose, as you would with a TSNE plot, the different transcript expressions, and you can see which region, which transcript dominates. So that was already quite illustrative. Yeah. Yes?
What are the labels? How did you get the colors?
So these ones, the cells have been really sorted at these different stages. Okay, so this has been pre-sorted, so it's sort of a bit discrete, but because they are very close by, I think they're just 0.02, like a quarter of a day or so, by distance. Actually, some of those cells are already there because they're just a bit further advanced, and so it's not as discrete as you want.
Sure. I mean, is there any interpretation of the purple, the putative endothelial, tracking all the way back into the, pluripotent? It's interesting. I mean, it goes pretty far down.
I mean, I don't know how many cells these are. You always have to be careful with the plotting, but right. I think this is all purple, right? And this is blue, I guess.
The purple extends all the way.
So I think there might even be a purple one, right? Yeah.
But there aren't very many red in that tail, right? So it could be that it's simply that you can see in this plot, right?
Yeah.
It is fair to interpret this as saying that the blood cells sort of differ more from the ancestral, I think, than the purple did.
So I wouldn't overinterpret a single.
Unless red was plotted first, then we just can't see what's underneath. Yes, that's a good point.
Yeah, so that's a very important thing. Whenever you do scatter plots, randomize before you plot, right?
That's a good point, yeah.
But also in this case, of course, you shouldn't, maybe if there's a single cell doing something different, you should not overinterpret. Obviously, you know, all of the cells, even if they were very noisy, would sort of have compressed on that structure. So maybe not overdo that. But I just want to point out that if you just do PCA, you see this dominant transition as well. So you see this thing here, but you don't see this splitting off very well of the endothelial part, which just clumps up here. And if you do something non-linear, such as t-SNE, which is very popular in the field, obviously, again, you can play around with perplexities or with parameters of that, but you see some type of splitting off of things which might not be actually doing much. And I think the reason for that being that t-SNE actually forces some type of clustering structure which you might not actually have in the data. Yes?
What about other linear approaches like ICA, which are more commonly used for trajectory analysis?
Yeah, this actually, ICA, what does it do for dimension reduction? It just does do a PCA and then just reorient the axis. So in fact, the ICA very much looks like that. So if you do...
The ICA looks like the diffusion coordinates.
No, sorry, like the PCA, sorry for that. Because, what does ICA first for dimension reduction actually does a PCA, right? And then only rescales the axis.
But it also identifies a more statistically independent signal. I mean, I don't know, maybe you'd have to look at multiple components. You would not necessarily see all the information on two components. I don't know, but I know that's how some of these other algorithms work.
So I used to do a lot of work on ICA before, and I think actually with ICA you could actually adapt the noise models maybe a bit more to the particular situation, but for dimension reduction, ICA is so you can actually show that if you just look for an independent subspace, then the PC subspace is always the optimal subspace to do first.
Subspace, but then if you're still finding non-orthogonal.
Oh yeah, of course. Right. So ICA won't basically do this, but would maybe do something like that or so.
Okay.
But it would just rescale the axis, but wouldn't look for a different subspace.
So by rescale, you mean kind of it faces the axes up with
exactly.
each other, but
only with each other, not with the other ones. So the subspace. So basically, if you want to do a two-dimensional ICA, it's always so it's equivalent to doing two-dimensional PCA and then doing the ICA.
And then just doing some invertible linear transformation on that stuff.
Yeah.
So is there also like a PC or an IC that well traces out those endothelial cells? I mean, is it just that you can't look at it in two dimensions?
Yeah, I think that so. I mean, clearly there seems to be one component dominating that. So there has to be particular factors, particular genes that only pop up in that are doing that. So you should see it also at some point in some downstream IC. Yes.
But if it's embedded in a nonlinear way, then it's not true that there should exist some linear reduction that captures that.
But we do know, right, that a few genes are just doing something here. So I guess you could come up with one axis. It's just not dominating in the variance here.
Locally or around one of those points,
down
but not globally, right?
Yeah.
It would just be lower down the PCA.
Lower
I was just, but that's totally possible. I was just wondering, I mean, one of the reasons why people use TMNE is because they don't want to have to pick how to visualize in two dimensions, like which subspace, right? And so, and I've kind of run into this problem with diffusion components, right? Where, you have to once you get a complicated enough set of trajectories, 2 or 3 or 4 or whatever, diffusion components are not enough. Do you have any way of approaching that yet?
Yes, so this is
visualizing early development once you have more than,
So the question is just as in PCA, right? What happens in higher dimensions? What's nice about t-SNE, such as similar to ISOMAP, well, a few methods that are sort of made to visualize as much as possible in 2 dimensions.
Right. And when diffusion maps are the right thing, but you have more than 2 components that are needed to really understand the.
So far in practice, we just go through the different projections and pick them up. But
you do the biplots. You just take pairs.
Or maybe 3-dimensional if you have a very good eye or something like that. We tried to buy a VR. I don't know if anyone of you is actually working in a wet lab, but we're always so jealous of the wet lab people, so we want to have something more. Yeah.
Wait, so did you buy the VR? I mean,
I think
we should equip computational labs with VR headsets.
Yeah, we did.
It should be standard issue, and we should basically,
So actually, I want the HoloLens, but this we don't have yet. Yeah. And it's also a good show if some funding people comes by, right? You can just.
Yeah.
So our solution currently is actually we do then the lineage estimation initially in the original paper it didn't scale so well to smaller components. And actually now in the new version I show code for that afterwards. Actually it's much better. So be happy if you try that, if you want to try that again. Yes.
One other thing is that it's not like t-SNE is really for visualization. Diffusion maps is finding structure, and the two-dimensional plots are beautiful to visualize, but you could also do subsequent analyses on the results of the diffusion maps. And that's unlike with t-SNE, that's actually a reasonable thing to do with diffusion maps.
But you know,
but just how do you? I mean,
but like,
how do you understand what you're finding in the? I mean, maybe you have a great, manifold in there that represents your data, but no biologist can look at it and tell, right? You need a way for people who know something about what they're expecting to be able to vet what they're seeing and understand the relationships there. Here, they show these maps exactly because people look at that and they say, oh yeah, that's what I would expect. It's doing the right thing. So what do you do when you have a manifold that's more complicated but still, represents something that should be vetted?
You might have seen from Alan Klein's lab this string-based embedding, so it's just really force-based embedding of the graph.
But they say the same thing. I mean, at least I didn't have those many from their lab, or they said, the force-directed thing you're saying, right? Yeah, well, they said once we had a complicated enough diffusion map, it was a mess and we couldn't use it either. So they only used it for sort of simpler. So it's really good when you have more than 2 but less than
I think it's good to pulling out the endpoint. So we have a blood example where this one actually worked out quite well.
Right, but the endpoints are really easy to see anyway. What you were interested in is the relationship.
And diffusion maps, you usually get them in different dimensions actually.
Right, that's true. Yeah, you could see them all, but if you want to see how things are related, which is what people are really trying to understand when they're doing a trajectory,
be happy to discuss you seem to have to look into that quite a bit. So I just wanted to point out one more thing. You said, of course, in t-SNE you shouldn't do any downstream analysis, but de facto state of the art in many papers still is where they actually do this, we call them fingerprints of the data, and then they compare them across different papers. You know about that, right?
There's so many problems with that.
So people really, I mean, I think in the first pipelines, they really did t-SNE and then k-means on the 2D t-SNE, and then you find the clusters, right? And boom, I find my cell types.
Me too.
Well,
so wrong.
Okay, so let me but the point I'm trying to make here is not only that you maybe find some visualization, but you also seem to be some type of ordering of the cells. So you might get an idea of cells at different temporal stages, and this is what the field maybe calls pseudotime, that you might get some idea of cells that are sort of maybe a little bit too early than where they should be, maybe some are later, so you get some type of within cluster and maybe also across cluster time. And this is maybe similar to when you stand at Harvard Square and you look at different people, you just get a snapshot, right? So you don't actually get any trajectory of the cells, of the people there. But if you just order the Harvard Square people by size, I guess you might get some idea of human development growth, or I don't know, something like that, right? So this type of evolution, I think it's called what the field calls pseudo-temporal ordering, and I think one of the first papers, a quite seminal paper, was put forward by Cole Trapnell, where he has been doing exactly that. There was a question on ICA dimension reduction, put each of the cells there, and then fitted a min-span tree through this two-dimensional space. There was actually an analysis done on that 2D space. But you could, of course, increase the dimension and so on. So the idea there was, just as before, because you now see the structure, let's fit a tree in that data that, as well as possible, explain this data now, and then fix maybe some type of cell, maybe just take the one that's sort of the two that are farthest away, call this beginning of pseudotime, and then call this index along this tree temporal state of that cell. And of course, there are all kinds of problems involved with fitting minimum of a cost function, right? It's obviously not so robust against noise, and there have been a series of extensions where you do bootstrap of that, you do a few more of those samples, but essentially you find this type of structure. There has been a later approach by Dana Peer, which wasn't done for RNAseq, but for Zeithoff data, so more low dimensional, and she called it very nicely Wanderlust. Are there any Germans in the room? Yeah. Even maybe not. So I was always fascinated how they come up with these cool names, because then she had a branching version which she nicely called Wishbone, which also I think a really cool name. But the idea there was that you again look at this graph, or this k-nearest neighbor graph, and then you say you want to transition from x to y, and you somehow have to do something against shortest path shortcut, so you want to have some sort of average behavior through the graph. So she came up with not basically doing shortest transition, but inputting a series of what she called, I think, landmark cells or something like that, and then sample
waypoints, I think she called
waypoints. They call it exactly, not landmarks. Sorry, waypoints. And then there was a sort of a double bootstrap approach where these waypoints have, I think, also been sampled. And then she constructs some type of average trajectories. Essentially, it's a generalization of a shortest path-based approach. And I think the trick, if you replace this one now by the whole ensemble of random walks, is that you actually do away with all of these shortcuts because you will actually look at all of them overlapping at the same time, which is what you do in the diffusion maps. Yeah. So that was basically our trick. So I
do want to hear about your pseudotime if you have the slide up.
Yeah. So basically the story here was there was no clear branching, and initially we developed this in parallel with Wishbone, so there was no branching at the time yet, and we wanted to actually upscale that. So what we did was now we looked at we wanted to somehow define a more robust manifold distance where we actually go along this line. So what we do is we construct, again, probability of jumping from some type cell X to Y. We call this MXY by looking at one random walk, but not at a single one now, or at one that goes through waypoints as you had in Wanderlust, but we look at all random walks of all different lengths. So not only the shortest one, which, the shortest one could make an issue. If you have some type of outlier cell here, you would go like that, and you would sort of destroy your temporal process. So we look at all at the same time, and we accumulate these now across the different lengths. And this you could maybe call some type of scale-free version. So it would actually also allow some type of going back and forth and so on. And then we take this new matrix M here, which says how probable it is to transition from X to Y across all of these different random walks. You have to take out, as I said, give a bit more detail about how we construct this in a minute. But just to get to this distance here on the manifold, now we compare columns of this M. Okay, so we say X and Y are close by if their transition to all other cells is very similar, and they're far away if it's very dissimilar.
Wait, so this is an integration across the time parameter in the diffusion, right? Like how long you've run the diffusion.
So it's not. So this is idea of mean first passage time. That's a bit along this line, yes. Actually. So in this case, we really accumulate all of these probabilities. So basically, what we do is we take this transition matrix here. We have to remove the steady state, otherwise it wouldn't converge, right? And then we say, okay, let me show it here. So we say we start at a cell X. So this means essentially just take the unit vector where there's a 1 at the cell X, 0, 0 otherwise. And if we now just apply the transition once, right, we get the probability to transition to the next state. Right? Now we don't apply it once, we apply k times, and then we just sum over all of those different k's, right?
Sorry, but okay, so if you remove the steady state,
yeah.
I mean, if you didn't remove the steady state, it's ergodic. It's going to basically every cell is collapsing to every cell.
Exactly. Then just get the density.
But if you remove the steady state, don't you still have dominance by the next eigenvector in? I mean, like.
Oh, yeah, it's true.
How
there's a decay. Like the first diffusion component dominates, and the next one dominates after that.
Oh, I see. So basically.
So it's a weighted sum of the different diffusion components.
I see. So as k increases, different eigenvectors will in some places in that.
So also
start to play the dominant role, and you're integrating across all of that.
Exactly. So it's like also for diffusion maps, we never show the zeroth component because it just visualizes the density, right?
Right.
And this is, I think, corresponding to eigenvalue 1 or something like that.
So the lower ones, as you raise
the lower
ones are decaying more and more. It's just more and more exponential decay.
And sort of the somewhat nice thing was, before Dana, for example, in Wishbone, she actually had to do samples of those to do even the shortest path with the waypoint thing to do that. But of course, this is all random walks. We know how these things behave. So it's just an infinite sum of a matrices, which is a geometric series, right? So we can rewrite this, in fact, in closed form, and we don't have to solve this. So it's really simple, but it turned out to be really useful for us. So we basically don't have to approximate it.
And this is related to the expected time that the random walk first goes from A to B.
I think so, but we couldn't exactly write it up.
It's sort of related to that.
Yeah, so we replaced this distance by mean first passage time, which is exactly that.
Right.
But it's not the same.
Oh. It just feels similar.
It feels, yeah. So I first proposed this one before Lale came up with that, and turned out this is more robust. So we use that. But what is nice, so this is basically just how we calculate the M, right? And then now we take difference of this M, and we weight it a bit by the zeroth eigenvalue, but essentially we now get a distance that tells you how far cells are apart on this high dimensional space fact, we want to interpret it as some type of geodesic distance. And I could now connect this a bit to how this is called to the actual evolving random walk on the thing, but I will actually skip that and just go to an interpretation of that, if you don't mind, because I am a bit over time. So, yeah, because I haven't finished, actually. I promised you not only to say that we have good distance, but that we can actually identify branching as well, right? So how do we do that? So there we start again from this x here. And we want to see if let's take two extreme cells, and in practice you can also find all kinds of heuristics to pick them up by automatically. But let's say we can ask the question: is Z branching off from X and Y? Then what we do is we start one random. We start our increasing DPT from X. We just measure distances along this line, and they would be increasing along this line if this was really proper geodesic distance. We do the same thing from Y, and what you see happens now is that along this main branch X and Y, these type of orders that start at the opposite ends are clearly anti-correlated, whereas when they branch off here, they start becoming correlated. What does it mean for order to be correlated? Well, you can measure this with Kernel's tau. If you remember, right, this was the difference between the concordant and the discordant pairs. So you can actually really measure that and basically get this jump region, and you do it from different times, so you get some type of region that's uncertain. You can actually determine an optimal way of splitting these off. Okay. And maybe as last point, whenever you write up these pseudotimes, I guess it might be important to think what time you can actually learn to start with. If you really have some type of cell type that is quiescent, is not doing anything, you should not actually start ordering it in any way, right? I mean, obviously, if there's some type of variance, you start at some range, you get some type of gradient, but it wouldn't mean much. So the question we had is, if we are there, of course, here there's clearly some type of pseudotime, but if you're in a downstream state, you should not actually really do that. So what we had as aim was to collapse all pseudotimes of a region of cells where they're not doing much into a single certain state. So we want to sort of shrink down those differences. And indeed, this DPT distance, because from all of them, if their transcriptome profiles are not very different, would not actually differ much, and this is, for example, in contrast to some other approaches that really look at distances between particular cells. So I guess what you could interpret this pseudotime as a proper definition, if you want, is, or as a getting close to proper definition, is that the pseudotime should be maybe the deterministic part of some type of stochastic process that rolls down here. So clearly, because we just have snapshot data, we cannot identify a single cell's trajectory, right? Let's say the cell might be sort of playing around up there before at some point it gets the activation energy to roll down this volume in the landscape hill. So we cannot identify the stochastic part of it, but this deterministic part, how far it's down here, we can. And this we did in a toy situation. So for example, what we did here, we had this simple switch-like model here set up where you have one toggle switch that essentially represents this thing here. We simulated with Gillespie type algorithm. If G2 becomes active, then it starts activating this downstream cascade here. So you see sort of a double switch here. And I only show you this part of this branching process where G2 wins and then G6 wins, okay? And here you see different realizations. So you see first this one goes on, and then with the stochastic waiting time, it sort of gets less and less infrequent the further down you have really exponential waiting times, then this G6 comes up here. So this clearly you cannot learn. So if you want to evaluate or compare pseudotime methods, you should not really try to get close to that, because clearly there's stochastic waiting times of going out of this meta-stable initial state. But if you normalize this thing here to the actual density in the particular state, this is what we call universal time. So we basically just normalize how long we are in this particular state. So we look at this time integral if you want, then you actually get this sharper type of transition. And this is exactly what this diffusion pseudotime actually starts to reconstruct. This is unfortunately something we can show, we can only just show experimentally. But this gives us indication that we get really something that's closer to sort of the stochastic, averaging out stochastic part along this transitions on that manifold.
What are you normalizing by in the universal time?
So we take the density of cells in a particular temporal. So now here we have actual time, right?
Actual time, yeah.
So we normalize by this actual time, and many are here. So that's because they rest here, so they're being averaged out. They transition through. There won't be much normalization. That's why this one is squeezed down, and this one just stays the same, right? So this is actually what, so this is the only thing that could be learned. This one cannot be learned, but this one you can actually show that could be potentially learned with or approximated with a method.
And just with that simple formula, you get that those two pictures are, that's real. That agreement is incredible.
Is the right side just
diffusion?
So this one has been, where we just take this data set here without taking the time?
Without the real time?
Without the real time, just random samples?
It's incredible.
And then you get that. But obviously, there's not a bit of a noise model, so that's why the pseudotime looks so nice, right? But we didn't add additional transcriptional noise on top of that, right? So it's really just this data set.
Okay.
But still, we're very happy. I was very happy when I first saw this plot. And now to wrap up, and then I'm done. Sorry for taking a bit longer. Let's just take this example again, and now decompose this part here. So now we can actually draw these lines automatically, because now, this we just do by locally averaging positions of cells in this pseudotime. You see this branching. In fact, we can actually decompose these now into these two branching regions here, where these trunk regions overlap, and here they now go to these two different branches. And we can also get into particular dynamics, but I skip over that. I just want to show you now this particular heat map. So this is what I showed you before, right? So this was this heat map that we got when we just sorted them by similarity. And now, just instead of sorting them here by similarity, let's sort them according to pseudotime. And then you get this type of map here, which is much easier to interpret. So here we have now the pseudotime, first branch, second branch, and along this line, because now we have some type of temporal axis, we can actually sort them by first up or down regulation. So basically, whenever you see a first big change, you can actually put them in a sequence. So you actually now see some type of a sequence of potential programs that are being turned on and off. And you can cluster them, and you can look at densities, and you can compare the different clusters here to the other one. I said at the beginning that we have big data, so I want to do this not for thousands of cells, but I want to do this for tens of thousands of cells. So we recently came up with a tool, ScanPy. We call it single cell analysis in Python. We basically re-implemented and also streamlined a lot of these analysis. This was done by a postdoc in the lab, Alex. It's quite efficient to use and really nice. And I just want to highlight that if you want to play around with this might be really more fun. For example, if you go to cell sizes of 60,000 or so, and these are really popping up quite a lot now in different studies, if you do this with classic, this is the cell ranger, so this is a 10x software that has been already made for that. It does take you some time even just to do all the pre-processing. If you take some more standard pipelines such as Surat, also actually it even takes much longer. But if you do this now with ScanPy, you really get down to regions that are, again, in sort of a time frame where you can play around with parameters and study this in more detail. And just to highlight this, it's really cool to have these many cells, because suddenly condition numbers are so small that you can really look at covariance structures and so on of that thing. So it's really worthwhile to play around with high cell numbers. And it's also fun for computational part. Yeah, and the tool is efficient. And it works for the 60K cell. And this is, I think that there was this one question for the diffusion map with the dimensions. So in fact, for this big thing, the diffusion map is really bad. And initially we weren't able to calculate it, but with this ScanPy, we can now do it. And here you see, for example, the first two components, you see some sort of dominant split here, which is something sort of nice in transition that you don't see so much in a t-SNE. And if you go to some higher dimensions, you really see some type of structure. So you can really look at branchings. And I think we're actually able to get to some multi-branching. So, I think there was a question if you can do this iteratively now with this sort of more robust toolbox, seems to be working well.
So the correlation, anti-correlation can be applied in a systematic kind of iterative
I think so,
fashion.
yeah. So you basically just go to a branch and they do it again. And initially it wasn't so robust, but he kind of cleaned that up.
Do you use the sparse computation techniques to get diffusion maps on
you
the
had to do
set?
that. Yeah. Okay. And in this case, it was still fast. But, and this is seriously the last slide now, this is one big 1 million data set out there, and this is something you cannot analyze. So even the other stuff you can't fit in R. This one, we're actually able to do it, and I think it looks really cool, right? So you can just do the t-SNE on that scale, and I think it took like an hour or so with the software, and we were also able to do the diffusion maps on this. So I don't even say what this data set is, it's just large, okay? but you see all kind of structures here, which I think are sort of put here somehow in the t-SNE. So I think in this case, it's actually really good on that scale of a data set not to do it in 2D. So I think that's really worthwhile to look at more fine-grained structures, and we're basically currently working on interpreting that. So with this, let me come to conclusion. I think I don't have to summarize, basically just said batch visualization pseudotime, and then I had some outlook which I maybe can discuss in person. These were the people involved in the lab and the institute, and thank you very much for your attention.